Welcome back to the Medical Microbiology Review. We now enter into the world of fungi. This is session number 5, Mycology number 1, the endemic and thermally dimorphic fungi. I have no financial disclosures. Here's my background. We now enter mycology for the next 3 sections. So we'll start off with our endemic, thermally dimorphic endemic fungi, starting with Histoplasma capsulatum. This is found in the Mississippi and Ohio river valleys as well as the eastern United States. It is endemic to these areas. It is most associated with bird and bat guano, spelunkers, so cave divers, and birdie attics. Here, this refers to people that go up to clear their attics and birds had nested in there and they clear it out and sweep away all the detritus, and it aerosolizes the spores or microconidia. Transmission is via inhalation of microconidia, although traumatic inoculation is also possible. With histoplasma, the immunocompromised, especially those with cell-mediated immunity defects, are at higher risk. So in other words, histoplasma acts more as an opportunistic pathogen than a true pathogen. Do not be mistaken, this is a true pathogen. However, it has a higher predilection to those that are immunocompromised in some way. Identification of histoplasma is largely based on morphology. So at room temperature, it grows as a filamentous mold, and at body temperature or 37 degrees, it grows as a yeast. Its mold form is characterized by tuberculate macroconidia, in other words, these spiky, big macroconidia here, and smooth, small, round microconidia that you can see here. Its yeast phase is characterized by small 2 to 4 micron oval budding cells that are commonly intracellular within macrophages. Antigen testing is available, and there is high levels of cross-reactivity with blastomyces. So here is another picture of histoplasma showing you the tuberculate or spiky macroconidia. This particular picture doesn't have very many microconidia, but that spiky ball is quite characteristic of histoplasma. Moving on to blastomyces dermatitidis. This is endemic to the Mississippi and Ohio river valleys, as well as the Great Lakes region of the United States and Canada. So there is great overlap between the distribution of blastomyces and histoplasma. Blastomyces is associated with decaying organic debris near waterways, and there is a high association with those that have outdoor occupations or outdoor recreational activities, especially hikers, and is more likely seen in adults than kids. One classic story is that somebody will go hiking with their dog, and their dog will develop this infection, soon followed by the owner developing an infection with blastomyces. This is not spread from the dog. This is just similar exposure, just different times and different doses. The animal being closer to the ground likely has a higher infectious dose exposure, but that is a classic story. So blastomyces acts more like a true pathogen, with transmission through inhalation of the conidia or traumatic inoculation being the most common. And identification, again, is mostly based on morphology. We're at the room temperature filamentous mold phase. You get a lollipop on a stick appearance. Here you can see this in the top picture, where you have smooth macroconidia that come off lateral conidiaphores. At the yeast phase and body temperature, you have relatively large, so this is 8 to 15 micron, round, thick-walled yeast that have broad-based budding. And here, broad-based means that it's roughly half the width or half the diameter of the cell. Another characteristic morphology here of blastomyces that you can see here in this bottom right picture is what is called nippling. So that's another cell budding off there, but not quite at the whole phase of broad budding. Blastomyces is negative for beta-D-glucan when in the yeast phase, so serologic testing for beta-D-glucan during active infection is not particularly helpful for diagnosing blastomyces. So here you see a prominent capsule of blastomyces, some broad-based budding coming off, and poor staining of that capsule. So this is an H&E stain from a tissue biopsy. Coccidioides immitis is found. It is endemic to the desert southwest of the United States as well as northwestern Mexico. Exposure is largely related to aerosolization of soil and dust. So think sandstorms, construction, those kind of things out in Arizona, Southern California. Transmission is via inhalation of arthroconidia. And this is generally considered to be a relatively high-risk laboratory exposure organism and should be handled underneath the biological safety cabinet hood at the bare minimum. Morphologically, Coccidioides in the filamentous mold phase has barrel-shaped alternating arthroconidia. You can see that here in this picture where the blue are the viable arthroconidia followed by lytic cells that easily break apart to form little wings that they can coast in the air once aerosolized. In the yeast phase, Coccidioides classically has a thick-walled spherule that is quite large, up to 120 microns in diameter, that is filled with small 2 to 4 micron endospores. And each endospore is capable of producing a new spherule that then fills with endospores. So here we have an H&E stain of Coccidioides. Here's a large spherule filled with endospores. Here are 2 other spherules with varying degrees of endospores filling them. Just as a review for the geographic distribution of our most common endemic dimorphs, you can see histoplasma in the brownish red color there, covering what is essentially the Ohio and Mississippi river valleys. You see broad overlap with the distribution of Blastomyces dermatitidis, again, Mississippi, Ohio river valleys, as well as the Great Lakes regions. And then you see the green Coccidioides immitis in the desert southwest of the United States. Moving on to Paracoccidioides brasiliensis. This is found in Brazil, Colombia, and Venezuela, as its name might suggest, and is associated with soil exposure and inhalation of conidia. Identification of this organism is largely based on its yeast morphology in tissue, where it has this classic captain's wheel morphology. So it looks kind of like the ship's wheel of an old-timey ship, where you have one mother cell in the center giving off multiple budding daughter cells surrounding it. Paracoccidioides is quite slow growing in culture, and it may or may not have conidia when it is grown in culture. And so using the mold phase to try to help identify it is really an exercise in futility. Talaromyces marneffii, formerly known as Penicillium marneffii, is found in Southeast Asia. This, again, is associated with environmental exposure, especially to soil and decaying organic matter, and transmission is via inhalation of conidia. The Talaromyces marneffii is largely an opportunistic pathogen, and it is highly associated with HIV/AIDS, especially those with the CD4 positive T cell count of less than 50. Historically, this has been considered an AIDS-defining infection. Identification of Talaromyces in the mold phase, you see fast-growing white to yellow-green colonies that have a diffusible red pigment. We can see that in the picture here. Microscopically, you see a classic penicillus or brush-like structure with smooth-walled 2 to 3 micron conidia. In the yeast phase, you largely see intracellular yeast that fill up macrophages. So here we see the penicillus of Telleromyces marneffii. Again, it's a paintbrush type morphology tipped with 2 to 4 micron conidia. Sporothrix schenki is associated with soil and thorny plants. The classic story here is somebody gardening in their flower garden, and they get stuck by a rose thorn, and now they have a cutaneous infection. This can also be a cause of respiratory infection in alcoholics. Most infections with sporothrix are actually in the immunocompetent, so it acts more like a true pathogen than an opportunistic one. And for identification in the lab, the filamentous mold phase at room temperature shows delicate septate hyphae. You can see this here on the right, that have bouquets or rosettes of thin-walled, teardrop-shaped conidia. Here we see this right here, or right here, with surrounding larger melanized or brown conidia in the milieu. You can see this here. Yeast phase is characterized by cigar bodies with narrow-based budding and the formation of esteroid bodies. So here we have GMS stain of sporothrix showing the yeast and some cigar type formation. The remaining slides are supplemental material to go over the clinical disease spectrum of these endemic and thermally dimorphic fungi. And that is all for this session.